everyone, and welcome to our webinar. I'm Ivy Elkins, and I am one of the co-founders of the EGFR Resistors, and I'm here with Jill Feldman, another of the EGFR Resistors co-founders, and Dr. Josh Baumel and Dr. Melina Marmarelis, both from University of Pennsylvania. We're going to talk today about progression and resistance as part of a three-part webinar series that is sponsored by Blueprint Medicine. Today's webinar is going to focus specifically on the topic, progression, now what? Necessary testing, common mechanisms of EGFR-dependent resistance, and possible treatment strategies. And we would like to thank Blueprint Medicine for sponsoring this webinar series for our membership. Let's get started. So first of all, Dr. Baumel and Dr. Marmarelis, can you both tell our group a little bit about yourselves, where you practice and do research, and how you're involved with EGFR positive lung cancer?
Yeah, sure. My name's Josh Baumel. As you said, I'm really happy to be here. I'm an assistant professor of medicine at the Perelman School of Medicine at the University of Pennsylvania. That part of the sentence is going to be very repetitive when Dr. Marmarelis goes, because we're both there. But in any case, I do research on patients with lung cancer, trying to improve outcomes for patients with lung cancer via clinical and translational research. I run clinical trials. I work with people in the lab. I do registry work, all with the overarching goal of trying to improve outcomes for my patients. I've had the pleasure of working with the EGFR resistors on many educational initiatives in the past, and really excited to participate today.
I will start the repetitive part of the process. I'm Dr. Melina Marmarelis. I'm an assistant professor at the Perelman School of Medicine at the University of Pennsylvania. I'm a medical oncologist focused in lung cancer. And I'm also the co-director of the Molecular Tumor Board, where we discuss a lot of the mutations in non-small cell lung cancer. I also have an interest in molecular testing in general and have a few initiatives looking at the role of liquid biopsy in improving molecular testing and the ability to detect an EGFR mutation. So I'm really happy to be here. Ivy and I actually met developing a clinical trial in EGFR, and Jill and I got to meet at a conference learning about all of the new initiatives. So I'm very happy to be here.
Yes, thank you both for being here. We really appreciate it. And we really are excited for you guys to share your knowledge with us. So the first question we want to ask is to you, Dr. Baumel. Can you give us a brief background on what it means to acquire resistance to targeted therapy? And specifically, when we're talking about EGFR, can you talk a little bit about EGFR dependent or on target mechanisms of resistance?
Yeah, absolutely. So when I think about this, I like to start, I have some slides here, which you guys can see, I think, walking through what a mutation is. Right? Because when we think about a mutation, people think of comic books and stuff like that, but really what a mutation is a change in a gene. And so what you can see in this slide, we have the blue and the red. These are chromosomes. Chromosomes are groups of genes. And you can have a mutation, meaning a change in one of those. So that change can be a insertion or a change. See how the blue goes from blue to green? That would be a change in one of the nucleotides, or it could be a deletion. See the red, we had previously had 5 rectangles, now we have 4, so one got deleted, and that could affect the function of the gene. The other thing which sometimes people hear about are these translocations. Translocations basically mean 2 chromosomes, they break apart, and they get stuck together wrong. So part of one gene gets put onto another part of a gene, and that can lead to confusing outcomes biologically. One of the most critical things we need to remember about these mutations in lung cancer is that the vast, vast majority of these are what we call somatic, not germline. So they are generally not passed through a family, though there certainly are family histories of lung cancer which are concerning. The other aspect of these mutations is they are generally seen throughout the tumor. So they're rather homogeneous. They're there. Every tumor cell has it. It sort of is its defining characteristic. So when we look at the normal conditions of what a mutation can do, and I think it just helps to give a perspective on how to view these on and off target alterations. When a gene is on, the light can turn on. It's just like when we have a light bulb. When the gene is off, it turns off. But when we have something called an oncogene, this is an on switch that gets mutated, and EGFR is an example of this. You turn it off, but it's what we call constitutively active. It's on no matter what you do. And so that is why we have drugs like OSimertinib, erlotinib, gefitinib, afatinib. These are drugs that essentially remove the plug from the wall, block the activity of that gene, regardless of what the gene is saying it should do. And that can lead to it being off, and we can see these remarkable responses. We also will sometimes hear about things like TP53 and RB. We sometimes talk about that when we're talking about small cell transformation in EGFR mutant lung cancer. These are what we call tumor suppressor genes. These are a little trickier. These are not an on switch. They're actually an off switch. They're the body's off switch to make something stop. And when those get mutated, a lot of stuff gets activated. And so as a result, if you pull the plug on that lamp, the lamp might still be on because of sparks flying everywhere. And that is a harder thing to target with a pill. The way that we test for this gets a little bit complicated. So PCR, polymerase chain reaction, this is the way we used to test for EGFR. It's a way to look at mutations. It actually, interestingly enough, uses an enzyme that was derived from a bacterium that was found in geysers. It happens to work really well at high temperatures. And so you take the DNA, you heat it up, then you cool it down, you heat it up, and you cool it down, and it allows you to identify these mutations. Fish, not the thing that swims in the water, fluorescent in situ hybridization. This is a way to identify gene changes, specifically often translocations. So what you can see in this picture is you see little green dots and little red dots. Those are the places where you expect to see those particular genes. And then you can see over here, green and red are on top of each other. That's not normal. That means that there is a translocation where it shouldn't be. So those genes are in the wrong place. Now, sometimes when we do fish, that actually makes a whole third color, but this picture doesn't have that. Never mind. And then IHC, it's another word that doctors like to use. We love abbreviations. Immunohistochemistry. Basically, this is paint by numbers of oncology. We stained the tumor for a specific thing. So the big one that people talk about often in lung cancer is PDL1, right? That's an immunohistochemical stain. We're looking at how much of the protein is present on that given cell. Now, of course, we talk about next generation sequencing. This is different from those other tests I was talking about. This test utilizes massively parallel sequencing, so I can look at not just one, not two, but hundreds, sometimes thousands of genes at the same time. This is what NGS is, which helps us tell a lot of information, but then the bioinformatics becomes complicated. I know that anyone who's looked at their NGS report gets a headache because you see all of these things on there. It can be very overwhelming, and your doctor is like, I only care about this line. And then you see the 20 other lines and you say, well, what about those? And we just don't know. The science hasn't caught up with that. And so that's something that's really important moving forward. And the other complexity that happens in terms of testing is DNA versus RNA testing. So DNA, that's your main genetic material. That's how your genetic material gets stored. But then it gets converted to RNA. Now, there's been a lot of discussion of RNA recently with the COVID vaccines. And so that's also using an RNA. RNA is the way that DNA expresses itself. It's the intermediary between your storage DNA and the functional proteins. DNA gets converted to RNA. RNA-based sequencing is a very good way to identify fusions and translocations. But given that we're talking about EGFR today, I'm going to go to the next slide. The other thing which people talk about when they talk about testing is liquid biopsy. This is a really cool picture that shows what liquid biopsy is. It's looking at circulating tumor DNA that's floating through the blood, and it's a way that we can detect these sorts of molecular abnormalities and do either NGS or PCR-based testing off of that DNA and identify molecular alterations. So all that testing is really important to get us to this point, which is that we now know that for patients with EGFR mutant lung cancer, the use of OSimertinib, which is a third generation drug, that just means we had first generation, second, kept getting better. OSimertinib was superior in terms of outcomes to the older drugs. And so this has become the standard of care for our patients with EGFR mutant lung cancer. And if we look at the side effects at the bottom, you can see that it has better survival outcomes, but it also has better toxicity, less rash, which is a big problem for patients. But when we give patients OSimertinib, the question is, what happens next? And so this was from some really exciting work that I was able to help participate in, where we looked at patients across North America, and we looked at their sequencing after OSimertinib to look at what mechanisms of resistance developed after OSimertinib. But what you can see here is a lot of stuff pops up. And if we see the light blue, that's only after OSimertinib. You can see things like MET amplification happen, or EGFR C797S, but you see lots of different genes, all these letters, it's a bunch of genes. And so the question that comes up is that some of these are on target, some of these are off target. What does that mean is an excellent question. So I have this analogy, which I think is helpful. So EGFR signaling, you can picture like a river. River's flowing. This is a blue rectangle. Let's imagine it's a river. So we give a drug like OSimertinib or any tyrosine kinase inhibitor, and it is specifically targeted to block that river. The river cannot flow through this rock. It's shaped perfectly. It fits in the water, and the water can't flow through. But we know that if you block a river, the water's got to go somewhere. And so on-target alterations, an example here would be C797S, is an example that, well, the rock gets a hole in it. So now the rock cannot effectively block the water anymore. And put into the perspective of the drug, OSimertinib can't bind to EGFR anymore. So EGFR signaling continues and the cancer grows. How about off-target alterations? So same situation we're starting with. We have EGFR signaling, that's happening, right? And we're blocking it with our tyrosine kinase inhibitor, with our drug. Off-target is when the cancer says, well, what if I activate this gene? I'd be able to grow. And so similarly, if the river can throw off some tributaries and amplify MET or have a fusion or have KRAS alterations, and all of this can lead to cancer growth. Now, I used the word amplify before. This comes up a lot in terms of understanding the report. And I want to talk about three terms that doctors like to use. And doctors love to use big words. It's probably one of my favorite things to do. So overexpression, gene copy number change, and amplification. So overexpression means that immunohistochemistry I showed you before, looking at the protein on the surface of the cell, it's more of it. So if we put this in perspective of you're walking up to a building, there's a sign on the outside, it says there's apple pie inside. So I might be lying. You don't know. So this is not very sensitive or specific. It's just sometimes it goes up, sometimes it goes down. Gene copy number change. Remember on that fish analysis I showed you, you showed how there was a certain number of each of the genes. You saw the green dots and the red dots. At gene copy number change, you have more of those dots. But that could be totally normal. That could be totally normal if you have more chromosomes you're measuring at that time. And so if you have a room and you say, there's 6 slices of pie being eaten, and you say, okay, that sounds like a lot of pie being eaten. And then you walk in, there's 6 people there. You say, okay, that's all right. Everyone has a piece of pie. That's all right. For amplification, though, those 6 slices of pie, let's say it makes 1 whole pie. Amplification is you're saying there are 6 slices or 1 whole pie being eaten inside of this room. You walk inside the room, you just see one very hungry person. That's not good biologically. And so MET amplification or other amplifications are the highest risk for the cancer cells growing as a result of this change, because the denominator, the amount of chromosomes, is out of sync with the amount of genes that we see. So yeah, I'll hand it back to you guys.
Well, that's a great explanation. I love the analogies, Dr. Baumel. There's some questions that and things that we wonder about on-target resistance. Is there any way or any biomarkers or clinical signs that can predict who's more likely to develop on-target resistance as opposed to another type of resistance?
I think that's a great question. It's a really difficult question too. I think the first thing to say first is by on-target resistance, we're talking about within the EGFR river that Dr. Bauml showed us. So things that are happening in that river. And the second thing is that it probably looks different depending on what EGFR targeted tyrosine kinase inhibitors used. So for instance, when earlier tyrosine kinase inhibitors were used, erlotinib, gefitinib, afatinib, the most common on-target resistance mechanism was development of a T790M mutation in EGFR. And so that's a very common type of on-target mechanism. Now that we have osimertinib, which is effective against EGFR T790M, we're seeing different kinds of on-target EGFR mutations. The most common being a C797S mutation in EGFR. And that can arise either with a T790 mutation or in the absence of a T790 mutation. And that occurs in about 20% of patients that develop resistance on osimertinib. So it is certainly prominent, but compared to about 50% developing T790M on earlier generation TKIs, it's still a smaller proportion. There is some data to suggest that patients with EGFR exon 19 deletions may have a higher incidence of developing C797S, but I think it's mostly, there are no good predictors for predicting who will develop this mutation. Also important in thinking about this is the position of C797S and where it is in relation to something like T790. And that has some implications as to how you can target it downstream. So Dr. Bauml, I'll let you add additional things, but those are my initial thoughts. It's a tricky question.
Yeah, I mean, I think that it's really hard to predict. And I think that the real holy grail here is not so much in playing the whack-a-mole game of treating the resistance alterations that pop up. It's trying to figure out why they're happening, right? So if this drug works amazingly and makes all the cells stop growing, then why do the cells grow eventually? So that, to me, that's really where we have to get to. And what we see when we look at these resistance profiles, where you're right, we don't see a big leading resistance like T790, is that it's going to be such small subgroups, and it's going to be really hard to develop drugs that work for everyone in the same way that osimertinib does, or erlotinib, gefitinib, afatinib does. So we really have to think about how we can change the approach to the overall management of this disease. And I think that probably means looking earlier in terms of novel treatment approaches.
Yeah, that's interesting you say that. I think about it in the same way, kind of like the river, as like if you're driving to a restaurant or to a house and there's a road blocked because of construction, well, what are the other paths to get there? And, you know, do we need to make sure all the paths are blocked if we don't want anyone to get there at all? It really, that's something we've thought about a lot lately. And I know you don't have the answers to everything right now, but in talking about these on-target mutations, what are the implications on appropriate treatments? And what about on the course of the disease?
So when we look at these on-target alterations, if we compare it to T790, patients who have EGFR-based mechanisms of resistance tend to overall do better than patients who have off-target alterations, for instance, with small cell transformation or something like that. They tend to have a more smoldering growth pattern. But that's not universal. And I think that what I will do is when I get this resistance testing, and I think that's really a critical point, is to get that resistance testing, I'll look at our panel of clinical trials that we have open. I'll look at clinical trials that are open in the region and try to identify the best approach for that individual patient. Like, I'm putting out to say, I'd love to be able to have a universal approach that makes everyone cured. That'd be amazing. But right now, what I have to do is use my best judgment to get someone to the best personalized treatment for them in later lines of treatment.
I think another thing I'll add there is that, at the time of resistance, when you get additional molecular testing and you find an on-target and, another EGFR mutation, that mutation is most likely conferring resistance to the current treatment. And so it does necessitate a change in treatment or an addition of something else, depending on what type of mutation it is. And so I think, we assume that although some of them are slightly different and we don't have a lot of information on a lot of them, that they are conferring resistance to the current treatment. One other kind of on-target mechanism we didn't talk about is actually loss of T790M. So it's not a new mutation, but it's the absence of a mutation that was present before. So that's another important thing to look for, and potentially actually something you could look for early on to herald resistance that's coming.
And do you see using CTDNA to kind of monitor progress? Is that something patients should be talking to their oncologists about yet, or where do you see that fit in right now?
So I don't use it routinely to monitor patients. It has been used in a research setting to look for decreasing allelic frequency, meaning decreasing of the amount of the initial mutation that was found as potentially a marker that treatment is working. Right now, I don't use it outside of the time of disease progression when I'm looking for a new resistance mechanism. There may be a time that we're using it more frequently, that we're monitoring or we're trying to predict resistance or preempt resistance so that we can add an additional agent prior to that happening. But right now, I don't have any indication that that is a viable strategy yet to employ this type of technology.
Yeah, the other problem right now with using serial liquid biopsies is that the liquid biopsy assays are simply not sensitive enough. The vast majority of our patients who are on a TKI clear their CTDNA. So you'll do a test, spend however much money, and you won't see anything, which is it's nice to see. Yeah, right. So it happens very frequently. Some tumors don't shed. But there are some newer assays that are in development that are hypersensitive. And I could definitely imagine that with one of those assays, yeah, it might even be better than doing scans. That is, to me, the future, but we're not there yet.
Right. That makes sense. Thank you.
Yeah, and that's one of the things, I like hearing about that, because that gives us hope for the future too, which is always something that as patients we like to hear. I mean, wouldn't it be great if we could be monitored with something like that as opposed or along with scans? It would be fantastic. But speaking of resistance, what is out there right now in terms of clinical trials or even, off-label treatments to either prevent this off-target resistance from occurring or to treat it after it happens? options. Could we talk a little bit about, what some of the options are for patients who might, either be concerned about this or experience this?
Yeah, so at this time, there's not anything that is standard of care to prevent the development of resistance. There are a couple of clinical trials in the first line that are ongoing that I think are interesting and relevant to this discussion. The first is FLAURA2. FLAURA2 is a study that is taking patients with EGFR mutant lung cancer and then randomizing them to getting either osimertinib alone, which would be the standard of care, or grafting on chemotherapy. And I know not many people get excited when I use the word chemotherapy, but chemotherapy is a highly active drug against this cancer. And the other thing about chemotherapy is it is not dependent upon a specific pathway. And so if you had cells that were relatively resistant to the osimertinib, those might be the same cells that could die when you give them chemo. We saw some data presented by a group in India, as well as a group in Japan, which showed remarkable efficacy when you added chemo to a drug like osimertinib. They were using gefitinib, but nevertheless, remarkable outcomes. That study is ongoing. Another study that is ongoing is trying to evaluate an EGFR MET bispecific antibody called amivantamab in combination with lazertinib, which is a very similar drug to osimertinib. That's being used in the first line with the logic that, as I mentioned, MET is a primary mechanism of resistance. And so patients are randomized to the combination or one of the two tyrosine kinase inhibitors to see whether you can improve outcomes. And it's the same sort of idea. It's trying to cut off a known resistance mechanism at the pass. In this case, MET is a mechanism of resistance about 30% of the time. So the idea is to try to prevent that resistance. In terms of treatment of resistance, when it develops, I will let Dr. Marmarelis take that.
Well, before I answer that, I think you bring up a really interesting point, which is, can you get rid of some of these resistance mechanisms that are maybe present at the time of diagnosis, and is that helpful in preventing resistance versus do these develop under the pressure of targeting EGFR? And I think it's an active area of research and a really important question about the biology here. So I think in addition to some of the trials you mentioned, we're all excited about two agents, Blue 701 and Blue 945, that have been basically designed to tackle this problem of prominent C797S mutation after TKI use. So Blue 701 is trying to tackle the problem of the original EGFR mutation with C797S, and then Blue 945 is tackling the problem of the original EGFR mutation plus T790 plus C797S. So all three mutations. And so we anxiously await the start of those trials.
What about, going back to an older generation of EGFR TKI, the first generation, second generation treatments? Is there any possibility in using those to treat off-target resistance?
Yeah, that's another great question, and there is some evidence to suggest you can do that. The problem is that most C797S mutations occur with T790 occur right next to T790, and when that happens, you cannot add a earlier generation TKI like erlotinib or gefitinib with efficacy. If it happens further away on the other copy of EGFR from the T790, then you can actually add erlotinib and gefitinib with some efficacy. So unfortunately, the majority of C797S mutations that co-occur with T790 are neighbors, are next to each other, and we can't use that approach, but we certainly need to be really diligent about making sure we know exactly where the mutation is and what the position of the mutation is, so that we have that as an option when we do detect it.
And the other thing to mention is that if we're using first-line osimertinib, T790 might not develop. And so if all you have is C797S, then in that first example you cited, I would often, in the absence of a trial, would just graft on erlotinib or gefitinib onto the osimertinib and continue both. That is not, there's no FDA approval for that, but that is what I would do in that situation.
So that's interesting when you talk about T790M being the original, the majority of people, the mechanism of resistance from first or second line was T790M. Now we're seeing C797S, and not as much, I get that. But is there research looking into ways to predict who will get these mechanisms of resistance so that up front we could treat accordingly? That would be ideal. Is that possible even?
Yeah, I think that it's really hard to do that. I think, is there research in it? Yeah, there's a lot of very smart people who are looking into this. Have we been successful so far? Not so much. There's some ongoing efforts to try to evaluate that, not with C797S, but Dr. Yu, who I know you guys had do one of these before, is running a trial where she's looking at if patients start out with TP53 mutation or RB loss, and we know those patients are at higher risk for developing small cell transformation, which is associated with a very poor outcome. Well, what if we came in with something earlier in that setting? That, to me, is our first glimmer of doing something like that. And as Dr. Marmarelis said, if you had the knowledge that C797S occurs more frequently in exon 19, well, then on the subgroup analysis of these upcoming trials, we could see, well, look, what about these patients at exon 19? Did they have particularly greater benefit? And the other thing which I will say about this is it's not our usual form of sequencing, but there is an ultra high sensitivity. That's the word that I'm using today, ultra high sensitivity. There's an ultra high sensitivity next generation sequencing that can be done to break down cell by cell to identify the genetic alterations in each one. And using that kind of technology, researchers were able to show that T790 actually preceded the introduction of a drug like erlotinib or gefitinib. And so that implies that the biology is predicted prior to treatment. I agree with Dr. Marmarelis that is not a slam dunk for everyone answer, but it implies that the biology does predict what happens. And I am hopeful that as the technology improves, we will be better able to personalize our approaches.
That would be fantastic. Yeah, sorry, Dr. Marmarelis.
No, I think Dr. Baumel and Jill, I think you also kind of highlighted this, but I think we've done a fairly good job of characterizing what other mutations are present at the time of diagnosis and what that might mean for efficacy of targeted therapy. But then it hasn't gone the step further that you're asking, right? It hasn't gone the step further to say, okay, with a TP53 mutation or a PIK3CA mutation, do those people have a higher likelihood while on a tyrosine kinase inhibitor targeting EGFR to develop C797S or T790M loss, various things like that. We don't have a good way of doing that. And so the trials right now, like the one run by Dr. Yu, are looking at just the efficacy and the presence of those mutations in the beginning. The other thing that I think is interesting that when Dr. Baumel was talking made me think of is, the association with exon 19 and C797S is in the context of T790. And I wonder how this will change as more and more people are treated with osimertinib in the first line, and that may not be, our numbers may change. And this is obviously a difficult thing to study because this is still a rare cancer. And getting samples to look at these serially is difficult, but I think it will be interesting to see how it changes over time.
Yeah, I do too. And as research progresses too, you think about, as patients, we want to have a strategy. We want to have a plan B. We want to have a plan C. And what we're doing is pretty much reactive now, right, in terms of treating. And we would like to be proactive. And I think I hold on to, I think everybody holds on to promises for liquid biopsy as well, because when you're getting a biopsy, you're not getting the whole tumor, you're not getting the whole picture, you're just getting a piece of it. And so we would like to get the whole picture. And that's where the promise, I think, lies within liquid biopsies, if they could be sensitive and, you know, specific for everybody. So that is hope. Do you have anything else that you'd like to add or say to our community? Anything that we missed? Or Ivy, is there anything you think we missed?
I think that one of the things that you said about having a plan, so whenever I meet a patient, I tell them that whenever I treat cancer, I like to do so with a plan, a backup plan, and a backup plan for my backup plan. It's a sentence that I always use. The difficulty, though, is that many people, and I group myself among these people, finds comfort in planning. And they've built their life around, I can plan this and I can plan this. And a cancer diagnosis takes away that power in such a large way. And it's something that we as oncologists need to be respectful of, the fact that this is like the amount of self-control that you can have in all other aspects of your life. And then here we're like, no, you got to trust us about the next plan. And it's a privilege that oncologists are in the position that we can help our patients, but we just need to be aware of how that is affecting them on a day-to-day basis. And we need to do better in terms of the research so that we can get to that proactive point that you're talking about.
So I just, what I have always told my team is I am not looking to throw the Hail Mary touchdown, right? Because odds are I'm not going to get it. I am just looking to get to that next first down. What we do with that series, we don't know yet. Mm-hmm. But what we do know is that first downs will keep us in the game. And the longer we're in the game, the more time research has to catch up. So, that's kind of more of a mindset, a different kind of mindset than necessarily having plan A, B, C, because we don't know if B will be B when we get there, right?
That's exactly right.
Mm-hmm. So, yeah, it's learning. It's an issue of learning to live with uncertainty and trying to, at least for me, focus more on what's going on right now and how I'm feeling right now, and maybe, staying in touch with potential options for the future, but also knowing that there's so much research going on, and, those options may be different and, have change something I can't even think of now, if and when I need them. So, when new patients come to me and ask me how I manage, the uncertainty, I always try to tell them to, keep an eye out for the future, but, just try to kind of go a little bit with the flow, because you don't know what's going to, come up in research.
And I think the rest of the world learned that during COVID, though, living with that uncertainty, not knowing when or what's next, right? So you all got a little bit of a taste into a person with cancer's world. So true, Ivy. Well, thank you both so much, Dr. Bauml and Dr. Marmarelis, for your time and for explaining acquired resistance to us in a way that we can understand. Love your illustrations, your the way you described it, Dr. Bauml, those were great. And thank you for your passion, for all the research that you're doing, and for fighting with all of us and for us, and your compassionate care. We really appreciate it. And thank you again to Blueprint Medicines for sponsoring this webinar series. It is so important that our community understands EGFR lung cancer and resistance, and we really appreciate the help with it. And thank you, CEC Oncology, for helping us coordinate these webinars. Have a good day.